Today we're going to discuss Chapter 12. Chapter 12 and 13 both deal with the immune system, just different aspects of it. So Chapter 12 is the beginning, and Chapter 13 is technically the wrap-up of the immune system. Chapter 12, the host defenses. Host defenses are a multi-level network of innate, or another way to say that is non-specific protections, and adaptive or specific protections, commonly referred to as the first, the second, and the third line of defense. Ultimately, we have different levels of defense for our body. The first is kind of the most simplistic, but it still helps defend us against invaders or foreign invaders. This is your concept map for both 12 and 13. It's kind of the overall view. As far as host defenses go, we have the innate or non-specific, and we have the acquired or the specific. With the innate or non-specific, we have the first line of defense, the second line, and then finally the third. Chapter 12 will cover first and second line. Chapter 13 is going to be what is going to cover third line of defense. This chapter we're discussing first and second line of defense. First line of defense is pretty much any barrier that blocks access to a portal of entry. POE is just portal of entry. So if you think back to the last chapter, a portal of entry is the specific way that an invader gains access to your body. If you have a part of your body that prevents that from happening, that's actually a defense against infection. It limits access to the body's internal environment. Literally, I'm not letting it get inside so it can't gain access. It's not really considered a true immune response because it has no memory and it has no recognition. As well, it is a very general action. It is not necessarily something that is specifically built to attack a specific invader. And when I say it's not specific to defending the body, our skin, for example, is designed to be protective, but that inherent design helps us defend as well against things like invading foreign bodies, whether that's viruses or bacteria. The second line of defense. The second line of defense is an internalized system. If I get past first, second is going to be the one that's going to try and defend me next. I've got protective cells. I've got protective fluids. We have processes such as inflammation and phagocytosis. This acts fast at local and systemic levels once the invader is past that first line of defense. So what do I mean by local versus systemic? If I cut my finger, the only thing that I need that to work as far as my inflammatory response and getting, protection to my finger is my finger. I don't need a full body fever for that. However, if I get the flu, I will get a fever and it's all over my body. I'm hot everywhere as far as that fever goes. For the cut on my finger, that's local. It just happens right here. It doesn't go anywhere else. If we're talking about a fever, I have 105 fever, then we're talking about a systemic reaction because it's my whole body. Third line of defense, this is specific and it has memory to provide a long-term immunity. The thing about specific immunity, the first time we get exposed to a pathogen, whatever it happens to be, the cold, measles, whatever, when we get exposed, we create an army to fight that specific thing. It's not for chickenpox. It's not for herpes. It's literally just for measles. That army stays with us. And if the measles ever comes back into our body, if it ever decides that it wants to revisit, we already have an army established from that first encounter. And that army recognizes measles, and that's it. So it's specific. And it remembers it. So if it shows up again, it knows it's my job to take care of it. First line and second line don't have that. Only third line has that. Something that's kind of interesting, but also makes sense. A lot of our lines of defense overlap with each other, and quite a few are actually redundant systems, meaning they're doing the same thing as another system is, or another function is. Combined, when you look at all of the possibilities that we have as far as defending our body, it really does make it a lot more unlikely that something will gain access. So if you've ever heard the term immunology, immunology is the study of the second and the third lines of defense. It's the body's response to infectious agents, and that includes things like allergies and cancer, because those two things are also abnormal invaders in the body. So, quick overview. The immune system is actually part of the lymphatic system. We have within our body 11 organ systems: the digestive system, the respiratory system, the cardiovascular system. The lymphatic system is what we consider as housing our immune system. If it is healthy, if everything is perfect and hunky-dory, that has 3 jobs. 1, it conducts surveillance in the body. It literally floats around, checking things out, making sure that things are either good, or if they're not good, it sends out a red flag. It needs to have the ability to recognize when something foreign is in the body. I need to be able to tell the difference between my cells that are normal and healthy and wonderful and, let's say, Staphylococcus aureus. If I do recognize something that is not supposed to be there, I need to have the ability to kill it, to destroy it. If it isn't there, it can't get me sick. So if I kill it, it can't make me sick. Our surveillance team, push comes to shove, is the white blood cells, or the leukocytes. Leuko means white, cytes mean cells. They form the surveillance team, and they examine cells in the body as they're floating around. And they can either identify the cells as being a self cell, it belongs in the body, it's supposed to be there, or as an invader, in which case it flips the switch to get them to try and kill it. That ability to surveil, observe, and recognize and kill is essential for your immune system to work. When this malfunctions, when I see self cells and I think they're foreign, so I attack them, that is actually called an autoimmune disease. Auto means self, meaning that my own immune system is attacking my body. So getting to how this happens, looking up here at the top, we have self cells. The orangey cells are the body's cells, your body's cells. See these little spike things that are on top? These are actually called antigens. It's a protein or a protein carbohydrate combo that identifies the cell as being you, kind of like a fingerprint. Your fingerprint determines that that hand belonged to you. Our white blood cells, these little dudes right here, are constantly floating in our body, looking at tissues and trying to find something that doesn't belong. But when they come to cells that have a normal self antigen, meaning that it actually belongs there, it floats off, it leaves these guys alone so they can live their best life. As long as you belong and are identified as being safe, they don't do anything to you. However, if you look here, this is when we detect something foreign. These little brackets that you see on the top of your white blood cell are called pathogen recognition receptors. A receptor is something that binds to something else. So we have these PRRs, And when they see something foreign on the surface of a cell, instead of being a self-antigen like this would be a foreign antigen, which your book calls a pathogen-associated molecular pattern molecule, or a PAMP. Once it recognizes that this is foreign, recognition of foreign substances, once it recognizes that this is foreign, it consumes it, and eventually it bombards it with so many enzymes that it destroys it. Destruction. Having the ability to do this is very important. I need to have some type of system in my body that can tell when something that isn't supposed to be there is all of a sudden found and get rid of it. And that's what these guys do. This is basically one of the most important parts of your immune system. The lymphatic system and the cardiovascular system. These two systems have very similar roles in the fact that they move fluid around your body. Cardiovascular system is designed as a pick up and drop off. I pick up food molecules in your intestine and I drop them off in your tissues so that they get nutrients, that type of thing. The problem is if I've got a pick up and drop off system, it usually means that there are holes so that things can get in and out. Well, we're talking about moving a fluid with stuff floating in it. That fluid, which is called plasma, sometimes leaks. And when it leaks, I end up with excess fluid around my tissue cells. When that fluid becomes part of the fluid around my tissue cells, we call it extracellular fluid. Extracellular fluid. So we have plasma when it's in the blood. When it gets in between the cells, we call it extracellular fluid. Now, if I leave it there, eventually it is going to cause damage to my tissues. Literally, it disrupts the balance of solutes and solvents, so salts and water, and it can actually cause damage to your tissues. So it's not feasible for me to just leave it there where it sits and collects and starts getting more and more and more as my cardiovascular system starts leaking more and more and more. So what do? Well, what I do is I have somebody else who picks it up. Like I said, lymphatic and cardiovascular are very closely intertwined, and one of the reasons is because when the cardiovascular leaks fluid, the lymphatic picks it up. Once that extracellular fluid goes from in between the cells to my lymphatic system in the tubing of that, it actually is considered lymph. So plasma, extracellular fluid, and lymph technically are the same fluid. They're literally just being named by location. There's some differences, but they're relatively minor. Now, because it harvests that extracellular fluid, and because it does this from everywhere in the body, anywhere there is a blood vessel, there is also a lymphatic vessel, meaning that I harvest from the bottom of the feet to the top of the head, to the tips of the fingers, to the center of my body where my liver is. This actually works perfectly as a good center for surveillance. If I'm picking up fluid from everywhere, no matter what, if there is anything in that fluid, I'm going to see it because I'm picking it up from everywhere. If I have a surveillance system that monitors the entire building and something happens, let's say in a, corner that nobody is ever at, but I have a camera there, I'm going to be able to see what happened, right? Same type of thing. I have tissue fluid harvested from all of these different places, and I can look at what's in it. So again, it makes a great headquarters for surveillance. It can also help us in that whole recognition and protection against foreign invaders. The lymphocytes, the phagocytes, and antibodies that you often hear about when it comes to our immune system, they actually kind of reside in this space. And again, that makes it very, very good as far as a center or a headquarters for this to work. We have two types of lymphatic organs, primary and secondary. Primary lymphatic organs are going to be the ones where we make the lymphocytes. Bone marrow is one, and that's where we make the B cells. The thymus is the other, and that's where we make the T cells. So the thymus is this little dude up here. That little dude up there is situated, if my heart's here, it's kind of situated up here. So it kind of sits above the heart. And when we have T cells, that's where they go to become functional. So when we talk primary lymphatic organs, we're talking about what actually makes our B and T cells. Where do they become active? Secondary lymphatic tissues are where those cells work, where they do their recognition, where they do their destruction, where they do their surveillance. So lymph nodes and spleen are two of the top kind of organs. The other things that we talk about are the salt, the malt, the galt, and the tonsils. And we call these lymphatic tissues. The spleen and the thymus are encased as their own independent organs. They are isolated from other tissues. The skin-associated lymphatic tissue, salt, the mucosa-associated lymphatic tissue, the malt, the galt, which is the gut-associated lymphatic tissue. These are little bundles of lymphatic tissue that aren't independent organs, but are instead embedded in other organs. And normally these places that they are embedded are potential points of entry. So having them in those places is a protective function. But it is still considered secondary because they're still doing surveillance, they're still doing recognition, and they're still doing destruction. And because of that, we do consider these little bundles that are embedded in other organs as a secondary lymphatic organ. As I said, our blood has two primary components: a fluid component that suspends everything, and then the solid component. Most of that solid component is going to involve cells. There are proteins and sugars and nutrients that are also floating, but for the most part, when we're talking about the makeup of the blood, we talk about the fluid portion, the plasma, and the cells themselves. So all of our cells and our platelets are derived from a cell called a hemopoetic stem cell. If you've ever seen the news, you know that stem cells are responsible for starting other cells. They're kind of like the beginning cell that gets all of the other cells made. We have two main types of cells: leukocytes. Leuko means white, cytes means cells, so these are our white blood cells. And we have the non-leukocytes. That includes the red blood cells and the platelets, although platelets aren't cells, technically. Red blood cells have one job: drop off and pick up. I pick up oxygen from the lungs and I go drop it off in the tissues. And when I'm in the tissues, I pick up carbon dioxide, bring it back to the lungs and drop it off there so you can go. But that's pretty much what they do. That's it. There's nothing fancy about them as far as, function goes. That's their job. Platelets. These little guys down here. Platelets are pieces of a bigger cell, and that big cell is called a megakaryocyte, and as it grows and we need more platelets, it tears chunks off itself, and those little chunks are the platelets. Now the platelets themselves serve several functions. They are most widely known for being involved in blood clotting. but they can be involved in inflammation with the chemicals that they can release, and they can actually destroy some blood-borne bacteria. But that's kind of the last thing people think of. As far as the white blood cells, we have two main types, granulocytes and agranulocytes. And literally the difference between them is how they stain. Granulocytes are going to stain with grainy looking patterns inside of them. Neutrophils, basophils, eosinophils, and mast cells are considered granulocytes. Neutrophils are very short-lived phagocytes that are present in our blood, and they eat and kill invaders. Basophils, eosinophils, and mast cells don't do that. They're involved more in the signaling part of our immune system. So basophils will release chemicals that help with the inflammatory response. They are the ones that, when signals get made, they start releasing chemicals, so your immune system goes, oh, wait a minute, something's happening here. I need to start the inflammatory response. Eosinophils, you will find these in high numbers when people have parasite infections, so hookworm and tapeworm, things like that. But as far as the function within the inflammatory response, they also release chemicals that help to kind of initiate and keep the inflammatory reaction going. Mast cells. All three of these float in the blood and are constantly moving around. One of the things that makes the mast cell unique is it is anchored in one spot, usually a spot next to a portal of entry. But as far as function goes, it will release chemicals that also promote the inflammatory response. But again, it's more specific because it's in this one spot instead of floating around the body. Our agranulocytes. We have primarily monocytes and lymphocytes. Monocytes are phagocytes that are found in the blood, but when they leave, they mature. So I'm floating in the blood, I'm a monocyte, I'm not doing anything special, but when you get me out of the blood, I transition into one of two types of cells. I'm either going to be a macrophage or I'm going to be a dendritic cell. Both of these cells are phagocytic, meaning that I eat things, because phago means eat. So, macrophage, big eater. They actually have a really high capacity for killing microorganisms and cleaning up dead cells. Dendritic cells reside in the tissues, and they're part of the mononuclear phagocyte system. So they have one nucleus, mononuclear, one nucleus. When they do consume something, they process it in a very specific way, and they become something that presents what they find to the lymphocytes, the other type of agranulocyte. You'll notice that I've got antigen presenting cell for dendritic cells, and I've got antigen presenting cell for my macrophages. These cells are going to process the invaders that they find. This is just a box, but I process it and I go to the lymphocyte and say, hey, look what I found. Imagine that there is a killer somewhere in your hometown and they know what he looks like. What are they going to do? Well, they're going to put wanted posters, right? When I have an invader that doesn't belong there, if I am the one as a macrophage or a dendritic cell that finds it, I need to show the wanted poster to the ones that will actually be able to kill it. In which case, we're talking about the lymphocytes. So our lymphocytes. We have T cells and we have B cells. I'm going to keep this very simple because we will talk about them in a lot more detail in chapter 13. T cells are involved in something called cell-mediated immunity, and T cells are assassins. They will either puncture holes into a cell or they will cause the cell to commit suicide, but either way, they're the ones that kill that cell. B cells, however, actually have a completely different job, and they're involved in something called antibody-mediated immunity. When they get activated, they divide into two populations, plasma cells and memory cells. Memory cells are exactly what they sound like. They go and they hibernate until a second infection happens. But the plasma cells become antibody factories against a specific target. So plasma cells make 2,000 antibodies a second. And if I make 1,000,000 or 1,000,000,000,000 of those, imagine the numbers that are being made as far as antibodies go.